National Monument. I'm Julie Huntington, Biological Science Technician, Glen Canyon National Recreation Area.
That's a lawnmower, AIS person for Glen Canyon.
I'm Mark Anderson. I'm the aquatic ecologist here at Glen Canyon.
Vanessa Glenn Lenore, biologist here at Glen Canyon.
Kristen Lewis, biological science technician, Glen Canyon.
And I Chief of Science and Resource Management, Glen Canyon.
And she worked with the Heritage Program with Bureau of English.
Shane Maurer, Bureau of Reclamation, Environmental Protection Specialist, Power Office.
John Steffen, Innovation Specialist, Southeastern Region, Subdivision Wildlife.
Nate Owens, AIS Coordinator, Subdivision Wildlife.
Something about
Erin Rainey, Aquatic Invasive Species Coordinator for Air Andy Silverwood, AIS Specialist for Air Management.
All right, that's everybody, I think. We've had a few of these. First of all, I'd like to thank the National Park Service personnel here at Glen Canyon National Recreation Area for hosting this meeting. And that's probably what we're going to try and do for subsequent meetings, is to try and team up as much as possible so we're not being duplicative, first of all. And for some of these, we're going to start giving them remotely. You know, from my lab and my office, travel funds are always, you know, getting increasingly harder to come by. But every once in a while, I need to get out and I need to see and meet more people. So that is the reason why we're here today. And it's a beautiful location, beautiful venue, and we thank them.
Sorry to interrupt, Dave. This is Scott.
Yes.
Can't hardly hear you at all.
I'm sorry, we'll speak up. If there's any way to turn this up. Is that better? Oh, it's in those microphones? Is that better, Scott? Scott?
Actually, be quiet. Let other people talk. Yeah, we can hear you.
If I had a nickel every time I was told that. All right, can you hear me now?
Yeah, it seems a little better.
Okay. I just wanted to talk about the goals of the Aquatic Invasive Species Task Force. We're funded by the U.S. Fish and Wildlife Service, but the primary goal of the task force is to, it's a multi-agency partnership spearheaded by the U.S. Fish and Wildlife Service, like I just said, to address the many threats, either active or potential, that invasive species pose to the Colorado River as a whole, the entire basin. It was also established to be a clearinghouse of information for many agencies, tribes, municipalities, private industry, and citizens currently working on or interested in the introduction, spread, dissemination, or overall effects AIS have on the anthropocentric use or ecosystem structure and function of the Colorado River. Yes, that's a very long mission statement, but it's still true. We look at many different species. Of course, quagga is always the 1,000-pound gorilla in the room, and we do discuss quagga quite a bit, but we also try and bring information about other potential problems from other AIS as well. We do have a website. There it is. It's updated quite frequently. It also has some reports on it, and there's a way that people can actually go in and report sightings. So it's there, and it's ever expanding. And if anybody has anything that they would like added to the website, just email me or get a hold of me and let me know. As far as meetings go, historically we've had 3 meetings, usually either in Yuma, Lake Havasu City, or Lake Mead. And we're still going to do that, but we're trying to cover the entirety of a very large basin at the same time. So that's one of the reasons why we're here today. AIS don't really know political boundaries all that well, so coming up into the upper basin only makes sense. And like I said, travel funding being what it is today, we're going to have some in-person meetings, probably at least 2 a year. and at least 1 or 2 kind of more remote meetings. Okay, we'll just be GoToMeeting because we have such good luck with GoToMeeting. But we'll have some remote meetings as well. It's always good to get out though and see people and talk to people. So with that, we have our first presentation, which is Jill Sherwood from Arizona Game and Fish Department. I can forward it if you need me to.
Yeah, Meg, I don't know, my voice doesn't carry very well, so I'll stand close to the microphone.
You got it.
I think we have to go back to the beginning.
Oh, sorry.
So, I'm Jill Sherwood. I am the HDMS data specialist with Arizona Game and Fish, and part of my position is managing the invasive species for Arizona and managing this IMAP invasive program. I've been with the department for all of about a month now, so this is all still pretty new to me. So please bear with me as I kind of go through this. It was my flash introduction to explaining IMAP invasives to somebody else, so we'll give it a go. So basically today I'm just going to go through, an overview of IMAP invasives, kind of let you know what it is, show you a few of the tools that are built within the actual IMAP invasives program, and just kind of walk through that. And, so it'll be a pretty brief overview. If you have any questions, please let me know. Get with me afterwards or anything like that. So IMAP invasives is overall, it's a network. And if you search for IMAP invasives, it kind of brings you to this site, which is really the launching point for the states that are involved in this network. Arizona's been involved for about 10 years now, starting with the statewide non-native plants, and it's kind of evolved from there and what's in our database. And as of now, there are 10 participating organizations. There's 9 states, Maine, Vermont, New York, Pennsylvania, West Virginia, Florida, Arizona, and Oregon, and then 1 Canadian province, Saskatchewan. And within, within each of these states, provinces, they have what's called a lead partner organization, or an LPO. And what this LPO does is they manage the state's invasive species data within IMAP. So that's kind of the go-to person for the state's invasive species data. And that also provides a pretty good network for any of the states that are involved. You can use other LPOs to kind of work and figure out how to administer your data. We have monthly meetings to work through tools and to try to make advancements for users and the functionality of the IMAP invasives tool. Kind of overarching that, there is NatureServe and the Nature Conservancy. They kind of oversee it, and then below that there's this FREAC, this Florida Resource Environmental Analysis Center, who provides all the IT support, the IT support for the tool.
So you said that was FREAC and not FREAK?
Yeah, I know, right?
Like,
Should be.
we missed that really good acronym.
Right, I know.
Just put in 1 letter.
I probably would have gotten slammed if I'd said that. So it's FREAC for sure. So the IMAP invasive project, or the IMAP invasives tool, it's really an online data management and mapping tool. And it's GIS based, so anyone with an internet connection can get to the site and use the tool. It doesn't require a lot of advanced GIS skills. It's built pretty user friendly, and it provides a centralized location for a lot of the state's invasive species data, and it pulls all that information together from multiple sources and standardizes it into a simple form that can be used by a lot of different people. It's visual, provides a lot of visual location, can be customized depending on management needs, and it helps with the early detection, rapid response, because all that data is there in one place. So a big part of this, and probably one of the most important parts, is it provides a platform for data sharing. So it aggregates all the data into one place, can be shared with different groups nearby. It provides a lot of collaboration, so it increases an awareness of invasive species, outside of your region, or what else is kind of happening within regions near you. It puts together a lot of different resources, so it actually increases the efforts that can be put forth towards, kind of attacking some of these occurrences. So who's it for? Pretty much anyone who has an interest in invasive species. General public can get on the website, they can view maps, it's a coarser scale. Right now it's county watershed. There was some talk of kind of, increasing the scale a bit to a grid size so you can see a little bit more what's happening without having a user account. Once you get a user account, you kind of move up to where you can view it on a better scale. So citizen scientists, community groups, they can work and contribute data. Any land managers, they can actually use that data, create land management programs. Researchers have access to current and historic data. And then you kind of tie it all together. You can have project leaders which take all of that information and have a project in one certain area.
Before we go on real quick, I just, if everybody online could mute your phone or mute your speakers on your computer, I'd appreciate it. It makes it easier for others to hear.
Yeah.
So in terms of getting a user account created for a citizen scientist,
Yep.
who decides on who is offered the user account, and what does that process look like? I mean, obviously you need to control that somewhat, so
Yep.
you control the quality of the data going in.
Yep, so that's me. I have the power. So that kind of ties into this. How IMAP is created, it's created with different user levels. So someone can go in, put their information in for a user account. And depending on how they're going to use the data, or what their level is, what their organization is, we kind of look at that and then assign them user accounts. So it starts at the 0, which is just public page, no user account. Goes down from there where you kind of start to get to the entering the observation data. So that could be at citizen scientist, community group level. Move up to assessment data, survey data. So that kind of moves it up a bit in what you're allowed, what you're able to do within that IMAP tool. So it controls it a little bit, just based on your user levels. And then the state administrator at the bottom kind of oversees all of that. And downloading data, once you get down towards these 8, 9 levels, that's where you have people that might need to manage data for their entire organization. So they would then get that level and that ability to download the data for their entire organization, but they can't for the regions or states or anything like that, unless you kind of move up then again in another level. Yeah. So I'm not going to get into a lot about the different methods for data collection, but I just wanted to show you all that there's different ways that you can collect the data, different ways that you can get it into IMAP invasives. We have some template field forms. If you're a pen and paper person, that allows you to kind of see the kind of information that you need to collect with IMAP to put it in there. There are also a couple mobile apps, IMAP Invasives, and then iNaturalist, and there's a project within iNaturalist for IMAP Invasives, and they have a little bit of different functionality. So you kind of get into it and decide which one you're more comfortable with. There's also the online data entry, which I'll touch on a little bit today and kind of show you that. And then there's also the ability to bulk upload. So if you have a large data set that you need bulk uploaded, you can give it to me, and then I'll work to get it in the system. So this is really the starting off page for IMAP Online. And this is what you'll see once you get a user account. And the things that you see kind of in the enter data, there's observations, surveys, treatments. Those all are different depending on your user level. So if you're at a user level, say 2, you're only going to see the observation. So those functions change a little bit depending on your user level. But you can enter data, you can view it, you can get it in a list if you want a list or table, you can query, and then if you have the ability to download, you can also download the data. And so this is pretty much the simplest form of data collection that IMAP starts with, and that's an observation. So this is a point data, a location of a particular species at a particular point in time, and it's really the who, what, when, where. 7 steps online to enter that data. You can submit photos, let us know who you are, what you saw, where it was, and then submit that via the online tool. And then once you kind of move up in that level, it gets to a few more of the advanced tools. And it's the assessment, which provides more information around a species observation, a survey, a more intentional survey of an area, the treatment, and the infestation management. So the assessment really captures the area around an observation. So you can enter disturbance, you can enter damage, you can enter intensity. And this is something where if you were just out there and you saw it and you wanted to survey the area, you could create a polygon around the area, put that information in IMAP, and it's stored as a polygon of that particular location. And then the more directed planned search is if you are going out and you're actually doing an intentional survey for a species at a particular location. And this provides a little bit more functionality in terms of what types of surveys you do. And you also enter some of the same information as you do in the assessment, but there's a few more things that you can enter into the survey information about that area. And then the treatment part of it, this is a function where you can go out to a particular location, say, draw a polygon around it, and actually, kind of whatever treatment that you applied to that, whether it be, fire, chemical, you can record that in a polygon area, so that will always be stored in IMAP. So you can see your observations, you can see your surveys, you can see your assessments, and you can also see which treatments are applied. And this could be over time or at one particular point in time. And what kind of ties all of those together, the observations, the assessments, treatments, surveys, is what's called this infestation management record. And this is where you can go in and you can enter information about it, but you can also say which observations are part of this infestation, which treatments were done on this infestation, and it stores it all in one localized location, so you don't actually have to go out and really search for that data. The other cool thing is that you can create a project. So if you have a particular area that you're interested in, you can assign someone or you can be the project leader, and this allows you to then track what other users are doing, so you're a little bit more in control of the data that's being collected, how it's being collected. You can also kind of track the time spent at an area on a particular project through this tool. And then IMAP also has a way to set alerts. So as a user, if you are interested in a particular area or you have your region, you can set an alert for that region. And if you want to know something, if an occurrence is approaching your region, so it's not quite in your region yet, but it's on the outside of it, you can set an alert if there is an observation in that area. And likewise, early detection, if there were a few occurrences kind of logged in your area, the alert would come up. And also if presence was established, if someone went out and found an established population, you would also get an alert on that. And then the system also has a suspicious distance report. And what this does is if someone goes out and records an observation that seems to be really far off from where it should be or from where anything else was seen, an alert comes to the state administrator. So I would get the alert, I can take a look at it, determine whether or not, is this a false report or is this an early detection, and then pass that information on to the different regions. So then you would kind of get the heads up that, hey, someone was out there, they saw this, just be aware of it. So it's kind of the, really quick overview of nuts and bolts of it. As far as Arizona's database, as it stands now, there's about 123,000 observations in there. Majority are bulk observations, aquatic and terrestrial, so there's about 219 species, 91 contributing organizations, and only about 13 projects. And because I just started this, I haven't really had a chance to look at the data and kind of try to figure out, where all of it's coming from and what it is. But once I get a little bit more into it, I hope to get a better idea and kind of break this down a little bit more to see where the data is coming from. Is it mostly older data or is it more recent data, just to get a better handle on the database. So with that, because contributions from users are what is really going to make the database successful and add to the strength of it. When I was going through this presentation, Dave had mentioned quagga mussels, so I went into the database to see, hey, what's in our database, and kind of looked around, and I've gone through, a few reports and found a few reports in some different areas, Canyon, Saguaro, kind of down by Yuma a bit. And then I looked in our database and we didn't have any points in there, so I went out and found this map, which actually has that data in it. So there's still some missing data in the database. So what I really hope, getting out here, and what I hope to do is to really connect with people so that I can get that data, we can get it in the database, we can get people sharing it, and we can actually utilize the power of the tool that it could be. So where are we headed with this? At the moment, IMAP doesn't have any ability to track pathogens. I know that there's been some talk previously about potentially adding that. I don't know where that is right now, but I know that that was one thing that they had actually talked about adding to IMAP. There's also a redesign coming. So you kind of saw the dashboard come up as it was right now. What IMAP Invasives is moving towards is more of the web GIS, so the ArcGIS online kind of interface, just to make it even more user-friendly and maybe be able to tie the app to a more collector type app or a Survey 123, so more of an ESRI kind of based app, so that people can work back and forth and make it easier to use in one platform versus trying to use the online, which is a little different than the apps. And then increased statewide participation is a big thing that I'd kind of like to do. So with that, if anyone has any questions, let me know. Yeah.
Yeah. So just, I guess, theoretically, suppose Utah and Idaho were both contributing to IMAP.
Yeah.
And as a state administrator, I wanted to research some of the Eurasian milfoil control efforts that Idaho is undertaking. Would I have the ability to search for those as a state administrator, or would that require something above that?
So I know in the east, because there are a lot more shared borders on Vermont and New York, I know they have a lot of communication between the states. I don't know for sure if you have that ability. I believe you do, but I don't want to say that for certain. The really cool thing about the LPOs is they all work really closely together. So as that state administrator, you'd have a lot of communication between neighboring states, and I know they do on the East Coast. It's across state lines, because stuff doesn't really stop at the state line. So I know there's a lot of cross communication. So I believe so, but yeah.
Any other questions for Jill? Any questions online? On the phone? Anybody? Is everybody still with us on the phone? Hello?
Oh, she muted them all.
Oh, we muted them.
That's what I was saying to you.
Sorry.
You have to
Making
do that.
fuzzy noises in the back.
Any other questions for Jill online at all? Anybody on the phone?
No,
All right.
we're on the phone, but not on a question.
What was that?
Questions?
No questions.
Okay, thank you. Thank you, Jill.
So thank you all.
I appreciate it. All right, moving on to What was I going to talk about next? Oh, a little bit about algal toxins and aquatic invasive species. I've worked on algal toxins for about the last 20 years or so, and I've given parts of these presentations before, but I kind of go back and rewrite them and put them together into what might be more timely or appropriate information to give at the time. So there is a large interest right now in algal toxins. So let's talk about that. One of the things when we talk about the ecology of what we're dealing with in the Colorado is we're dealing with the ecology of constructed ecosystems, all right? So it's difficult enough to figure out the ecology of more natural ecosystems, of which we're still doing, but most of these systems are quite young, and we don't know exactly what kind of species they're going to hold, what kind of niches are available, what kind of habitat's available, and that'll change over time. But we're not quite sure necessarily what. Thank you. We're still not quite sure what's going to move in, right? Or what kind of trophic states we're going to have, what kind of, like I said, niches might be available in the future for new AIS to move into. We're also talking about algal assemblages, primarily phytoplankton in reservoirs, right? Well, phytoplankton in reservoirs is one of the most environmentally patchy assemblages known to exist. So trying to sample for them, trying to interpret that data, and then trying to determine a long-term trend is really difficult, especially for phytoplankton in constructed ecosystems. Just a few things. I know I might be preaching to the choir a little bit, but that make the southwestern U.S. reservoirs different than north temperate lakes, right? One is certainly decreased residence time. Right? Our water doesn't sit in our reservoirs the same amount of time that it sits in places like the Great Lakes, that's for sure. Increased strength and longevity of thermal stratification. We live in a very warm area of the world. Incoming sunlight is not lacking. So we do have thermal stratification sometimes into December, January. It can last late into the year. Well, a lot of north temperate lakes can use those cooling waters as the way to decrease primary production. We don't have that here. Especially things like increased suspended sediment loading. I've worked with Paul Grimillon and some other people at NAU, and we've done sediment coring in the reservoirs before. We've gone back and age dated the cores to look at trophic statics changes over time. And trying to talk to people, I have people in Vermont, people in New Hampshire, and we talk to them about, our inloading of sediment. They ask me how many millimeters a year we can expect. We can expect a half meter during an event. Okay, so it's a completely different world here, and it's a world that they're not quite used to. We have cored to the bottom of Havasu before, and that was almost a 3-meter core. So it's deep. Those sediments are deep, and obviously that has a limnological effect. And of course now we have quagga. Very efficient filter feeders, obviously. High clearing rates of the water, high particle retention rates. In other words, we're going to take it in and we're going to hang on to it for a while. Can consume particles as small as bacteria plankton, right? Just a few microns, not very big. Especially things, well, we'll get into cyanobacteria in a little bit. Tricenids in general promote cyanobacteria through the process of selective rejection. It's been proven time and again. I mean, they can, right? What that means in a huge reservoir is a different story. It's dependent on the number of quagga that are actually there versus the overlying water. They indiscriminately filter phytoplankton from the water. We usually reject those toxic strains of cyanobacteria, right? Because of we don't know. We can't ask them if it tastes bad, but we're assuming unpalatable taste or size. Okay. So they've evolved these mechanisms over time. Quagga have been around for a very long time. Cyanobacteria have been around for 4 billion years or so. So they've figured it out. But I guess the big question is, can quagga alone cause cyanobacterial blooms? You know, if we have a reservoir or a lake, and you know it's basically a oligotrophic, and we put quagga mussel in there, is are quagga the only thing that's going to cause a cyanobacterial bloom? Probably not. It's a factor, right? But it's usually a combination of different factors that cause, well, eutrophication in general, but certainly things like cyanobacteria to bloom. Okay? One of those is some of the things that we were talking about earlier, incoming sunlight, but certainly nutrients is another, and nitrogen and phosphorus. In our watersheds, generally, in the Colorado River Basin, are usually what we would consider urban poor. I mean, there's not a lot of things that are happening, although they are, I'm being very generalistic. We don't have the populated watersheds like you do along Lake Erie. Okay? There are no large cities that are entering treated wastewater into the streams. So our sources of nutrients are coming in off of that suspended sediment like we were talking about earlier. That suspended sediment usually contains biounavailable forms of nutrients, reworked in the lake eventually over time. I've worked with ADEQ a lot on reservoirs in Arizona. We wrote some narrative nutrient criteria, and what we found, a lot of cases where we have this relatively low nutrient but high chlorophyll situation. Low nutrients are relative, right? But lower than what you would expect or compared to a north temperate type of lake. Sometimes we have high chlorophylls. Those high chlorophyll levels are kind of fleeting. They don't last that long, okay, because we have other controls and low nutrients. It's important that we keep low nutrients in our reservoirs. Okay? And what we find a lot of times are algal species well outside of what their established environmental range should be, environmental tolerance range. We find algae that are in waters warmer, colder, more saline, you name it. We find them well outside of those established regions anymore. It used to be believed that cyanobacterial blooms would only occur in eutrophic, maybe mesotrophic type of reservoirs or lakes. Right? It's not necessarily the case anymore. Sometimes we see these monospecific blooms of cyanobacteria in reservoirs that have that low nutrient content, but the biodiversity of the overall phytoplankton is super low. Most of these are very monospecific type of blooms. So we have microcystis in places that we wouldn't necessarily have expected them before. What quagga's role is in that is still up in the air. Okay? Lake Havasu, post-quagga, routinely experiences persistent microcystis outbreaks. 2014, 2015, Meade, Mojave, Havasu had I call it a bloom. Other people will argue with me that it wasn't a bloom. They certainly had a higher incidence of microcystis in those 3 reservoirs than they had previously. Okay. It persisted especially long in Havasu. One of the things about cyanobacteria is the abundance of biomass is not directly proportional to toxin production. Just because we find a potentially toxic species of cyanobacteria out there doesn't mean that it's actually producing toxin. And in some cases what we find is that we'll find the toxin itself far removed from where the bloom is. Sometimes even after that bloom has died back, especially after that bloom has died back. Okay? So it's really hard to tell. If we're out there and we're doing algae counts and IDs and we happen to find bunch of microcystis or anabina or anything, it's really hard to predict if that's actually producing toxin. We actually have to analyze for the toxin itself. In Havasu, there have been hits of microcystin well above the WHO recommended guidelines of 1 milligram per liter. And EPA and other people are looking at assessing limits, currently don't have any, for microcystin. Okay? And I get it, microcystin is the most ubiquitous of all the toxins. It's one of the most persistent, environmentally persistent, sticks around for a long time. But it's not just microcystin. I had a postdoc and I stole some of his slides for the next slides coming up. There was this paper, State of Knowledge and Concerns on Cyanobacteria Blooms and Cyanotoxins. It's just an overview of everything, these toxins in particular. Sorry for the propaganda, I didn't feel like reformatting his slides. So just to go over a few, there are dermatotoxins, or things that cause skin to itch. Right? Ecclesiotoxin, lingiotoxin. Of those two, lingiotoxin is the most common. Sores, itch, that kind of stuff. Okay. And he was a little hyperbolic in some of his health effects. Usually it just kind of comes and goes and people go home and they scratch and the next day it's gone after they've taken a shower. Some of these persist and can cause rashes. It depends on the person. Right? Some of these can persist for very long periods of time in a person. Hepatotoxins, things that affect our renal system, specifically microcystin, but there's also nodularian, and cylindrospermopsin. Like I said, most of these, of all these, the ones that we really know the most about is microcystin. It's the easiest to analyze. But at the same time, just because microcystin is below detectable limits doesn't mean there aren't other agrotoxins in the water. Right? Yeah, it's a little hyperbolic. Gastroenteritis, and then we go straight to death. So there's a lot in between there. Right? We're not going to just die. Carcinogenic, mildly so, very mildly so, and it would take chronic persistent doses of microcystin. Okay? The big thing with microcystin is usually that gastroenteritis. And it's also things like wildlife and domestic animals. Dogs don't know green water is green, we love swimming in it, and we're just going to drink it. Same thing with cows, deer, all sorts of things. And there have been documented cases of things as large as cattle dying from microcystin, especially domestic dogs, because that's what we hear about the most. There are some neurotoxins, I like this, paralysis, death. There are some neurotoxins. Anatoxin in particular, anatoxin AIS is even more rare. We have found anatoxin in some southwestern Arizona reservoirs. The thing about anatoxin is it breaks down with sunlight and water hardness, or calcium in the water, which is not lacking here. So we have some anatoxin. We actually have to go out there and freeze our samples in Dewar's flask immediately just to measure it. But it's not generally a problem. It is produced somewhat in very low doses, but at the same time it degrades very, very quickly. There is this BMAA, and it's relatively newly thought of. It's not necessarily newly found. The thing about BMAA is it could be as ubiquitous as microcystin. All species of cyanobacteria, from what we can tell so far, produces BMAA. Right? It is a neurotoxin. It can biomagnify and bioaccumulate. Yeah, potentially involved in some neurodegenerative diseases, but the risk associated with that, not many people are just drinking lake water. Okay? And I kind of alluded to this earlier about this environmental patchiness. It's difficult to sample for some things. Difficult to sample from reservoirs and lakes. We call that sample representative. Okay, it's a 1 liter sample representative of Lake Powell. It's the best we can do at the time, absolutely. And in some ways it is representative, but it's due to that patchiness that it's very difficult to capture that variability. Sometimes we have to do this in situ concentration. It's especially true for small plankters, things that are, might go unnoticed otherwise without that concentration. So we worked on this for a little bit. Yes, this is a shameless plug for something we made, but part of the problem with concentration techniques, and especially towing plankton tows through the water, is that you have one mesh size that you use at a time, right? So if you have an 80 micron mesh, you're capturing everything 80 microns and smaller, right? But a lot of things, especially as these nets start to plug, and they do pretty quickly in very productive reservoirs, you'll get things that just go around the outside of that net. So they're very specific. You're missing a lot just due to size fractionation of things that we find in the water. We find all sorts of different sizes of phytoplankton, anywhere from just a few microns, like microcystis, all the way up to some pseudofilamentous things that can be a few hundred microns, or visible with the eye. So we go basically from bacteria size to things that we can see that we're trying to capture. And they've created all sorts of, it looks like the Batmobile type of thing for plankton sampling. Very creative things. So quantitative versus qualitative, right? Qualitative, we just use as presence absence. We see if something's there or not. We don't necessarily try and count it. Quantitative, we count it. And the reasons we do that over and over and over and over again is to detect long-term trends. It takes a lot of data, especially in environmentally patchy populations. So that determination of sampling device is very important for quantitative studies, because we need to capture them as best we can. So we're always having this internal argument of determination of what best mesh size to use. Okay? Nets often quickly clog. There's no in situ size fractionation that we're doing. Sometimes it's difficult to determine the flow rate or the water that's actually gone through the net. So it's difficult, right? Capturing those representative samples. That in situ size fractionation can lead to things like earlier detection. And that's always what we're trying to do. Whether it's cyanobacteria or whether it's things like very small veligers or even trochophores or quagga. We would be able to find things like that if we had a better way to size fractionate things in the field. So we made one, which looks like a rocket. Basically, we kind of stacked nets. Largest net in the front, 250 micron net in front, 80 micron net behind that, and then a 10 micron behind that. And it works pretty well. You drag it through the water. There's flow nets in front of each net, so you know the exact amount of water that's flowed through each net, and you have a better calculation rate of what's gone through them. And it's portable. It comes apart. It's hinged, and you can put different net mesh sizes in each one depending on your study. You can also do vertical profiles. If you wanted to just drop it off the side of the boat, pull it back up, you could do that. And it planes through the water very nicely. Initial results from Havasu, yeah, I mean, we did get some size fractionation of things. So that size fractionation is kind of important, because if we were to use our 250 micron mesh net as just a single net, we wouldn't have captured those other things below it, or vice versa. So it's a better way to capture things that we find in the water. Just to summarize real quick, contribution of quagga to anecdotal evidence of increased cyanobacterial blooms is still unknown, right? I mean, there's been people saying, well, we've never noticed microcystis as much as we have now, and gee, it just happens to be after quagga. And we don't know. I mean, that could be completely circumstantial, or it could be real. But the bottom line is, they can't help. Right? It's been recorded and documented in other places, especially the Great Lakes, that yeah, there is kind of an exclusion of other algae post-quagga invasion. Climate change, yeah, climate change will definitely play a role. And you know, as we get warmer water throughout the year. And I did want to say that most of these algal toxins are easily removed through conventional treatment processes, water treatment processes. It's not a problem with drinking water. It's a recreational problem. And aquatic wildlife. And terrestrial wildlife. Toxin production is not synonymous with cyanobacterial abundance. I have samples waiting for me back at the lab now I have to look at, but apparently, you know, I've been running microcystin and doing biovolumes of microcystis along with that for years, and there is no direct correlation. Sometimes microcystin levels way up there and there's hardly anything in the water, and vice versa. Okay. In situ concentration techniques probably help. We used to be able to get more representative samples and to assess those very low concentrations. You see low concentrations of things, at least it advises us that, all right, this could be a problem on the horizon. And that's it. Any questions? Yes, sir.
So do you know what, like, you say the toxin production is not synonymous with cyanobacteria abundance, so do you know what triggers them to start producing toxins?
Oh, that's a good question. That's a really good question. For some things, it's a allelopathic. It's other algae. Right? So if you get growths of other algae around them, and I actually had a grad student who worked on just that, allelopathy. They are prone to produce toxins. I worked with Cylindrospermopsis for a while, and it produced Cylindrospermopsin when going from a high nitrogen situation to a low nitrogen situation. There's things like grazing. So see, that's just it. It's very, not necessarily species specific, but there are many things, and it's a combination of factors. Definitely grazing, definitely things like allelopathy from other algal species, of which they do have a tendency to produce toxin and get rid of other ones. And especially zooplankton grazing. Things like microcystis do affect things. I mean, the whole trophic cascade of, you know, if we have quagga in a water body, and let's just say they happen to at least promote a cyanobacterial bloom, probably not going to find the zooplankton that we used to find, which eventually might affect fisheries. So,
Do you know if, you know, one of them starts producing and then the other one sniffs it and it starts producing? Is that kind of a chain reaction? Once one starts producing, the other ones around it start producing as well?
it's usually because a lot of these things exist as colonies.
Right.
It's usually most of the entire colony that's producing. There's also intracellular toxin production and extracellular. Sometimes we'll find after a bloom starts to die back, that's when we start to find large amounts of toxin in the water, because everything that used to be within a cell is now out. For example, Lake Pleasant, they used to release, they have 2 gates on Lake Pleasant, which empties into the CAP. Scott, correct me if I'm wrong. But they used to release from the top, right? And then lake level would lower, so they would just kind of automatically switch to the bottom. And there was about a 15-degree C temperature change between the top and the bottom. So it lysed all this paraphytic cyanobacteria going along the side of the CAP canal. People will call up with mass complaints of MID and geosmin or taste and elusive colon. So it is one of those things where they start to die back. I don't think, we have measured individual cells of cyanobacteria producing toxin. We've measured whole colonies. I don't know if there's any signaling in between, other than that they share common environmental variables. Does that make sense? Any other questions?
Folks on the phone are unmuted if you have questions. No.
No? Okay. Thank you. Denise? Hello?
I just muted everyone again.
Oh, I'm sorry.
The green
I'll have to unmute it for her presentation.
Denise is online. Can you hear?
Yeah.
Okay.
All right, hang on, Denise. Let me get your presentation going.
Everything?
All right. And I'm pleased to have Denise Lindsay from Corps of Engineers, research biologist, talking about something that I'm very interested in, the genetic structure of quagga mussel in the Southwest. So thanks, Denise, for joining us, and I am ready whenever
Thank you for having me.
ready whenever you are.
All right, I'm just going to ask you all to bear with me as I have a coughing fit. I'm getting over pneumonia, so if I interrupt myself, please also feel free to interrupt me if you have questions or need clarifications on anything. Can I control the screen, or do you need to do it, David?
I can do it, or I can give you control if you'd like. Either way, whatever's easiest for you.
It's fine. You can stay in control. Okay. My name is Denise Lindsay. I'm a research biologist out of Vicksburg, Mississippi. I work in the Environmental Laboratory, which is part of the U.S. Army Engineer Research and Development Center. Today I'm going to be summarizing a collaborative study between ERDIC and USDR on the population genetics of the quagga in the southwestern United States. Okay, next slide. Interest in this project arose due to the unique water quality of each reservoir along the lower Colorado River and the possibility that some quagga populations might be adapting to those variable conditions. We use microsatellite genotyping to investigate the current genetic status of quaggas in the southwestern United States because understanding the genetic variability among quagga populations may help predict future distribution patterns and potential genetic control points along the Colorado River. Next slide.
Go. Okay. There we go.
Okay, so why will we use genetics as part of invasive species monitoring? And the reasons are basically the same as our desire for genetic health data of threatened and endangered species. Population genetics can provide a slew of information, including population size estimates, which are similar to census, effective population size, which is essentially the estimated number of breeding pairs actually contributing genetically to the population, overall and pairwise genetic differentiation between populations, genetic diversity indices, which can include allelic diversity, heterozygosity, and inbreeding coefficients, the estimated number of migrants into the population, and whether any genetic bottlenecks have occurred in the population and the time since a bottleneck event. This genetic data can then be combined with other factors to get a better understanding of the big picture and potential reasons for genetic variability and possible adaptations of specific populations. Next slide, please. There have been several studies that have analyzed the population genetics of quagga mussels in North America and Europe using microsatellite markers. The number of loci that were used and the number of individuals that were analyzed has varied greatly, with as few as 3 loci and just a couple dozen individuals. These days, microsatellite studies are generally not accepted or published without at least 10 loci and as many samples as reasonably possible, depending on the species. What we determined through our review of these previous studies is that observed patterns in pairwise genetic differentiation between populations and levels of heterozygosity when they were reported varied greatly. In general, the genetic diversity of quaggas in the southwestern U.S. Is reduced relative to the Great Lakes population. And most of the Great Lakes populations, excluding Lake Ontario, are genetically similar to the European population. However, the southwestern U.S. populations are more genetically similar to Lake Ontario. Next slide.
Did you get it? Did that change for you?
I got it.
All right.
Okay, in order to build on those prior studies, the main objectives of our investigation were to determine whether lower Colorado River quagga population genetics have changed over the past 5 to 10 years due to continuing expansion and establishment, and we wanted to use a larger suite of microsatellite loci for our analyses. Our plans were to determine linkages between populations, discover the degree of isolation of populations, identify any potential expansion pathways, and determine any genetic relationships with water quality measures. Next slide. Obtaining genetic material from quaggas, our field collections were taken care of by Sherry and Jackie. The plates were deployed and samples were collected, and then the samples were shipped to our lab to their lab on AIS. Next slide. We collected 30 individual quagga mussels from each of 6 reservoirs along Arizona's borders of Utah, Nevada, and California. From the lower Colorado River, we sampled Lake Powell, Lake Mead, Lake Mojave, Lake Havasu, Senator Wash, and Imperial Dam. Next slide. These 180 samples were stored at -20 until the extraction process was started. They were processed in batches of 12, and we did just 1 reservoir at a time in order to reduce chances of contamination or mislabeling. Mantle tissue was excised with sterile scissors and tweezers, and the extractions were completed with QIAGEN's DNA blood and tissue kit. The DNA was finally eluted in a volume of 50 microliters, and a nanodot spectrophotometer was used so that we could assess the quantity and quality of DNA prior to running further analyses. Next slide. We used a subset of samples to initially assess the 12 available debug and DBU microsatellite DNA loci that were developed by Wilson et al. And Feldheim et al. We then selected the 10 best loci for use with our entire data set. We would have loved to use all 12, but they did not all amplify DNA reliably. Polymerase chain reactions were then run across all 10 loci for all 180 samples. Microsatellite genotyping was conducted on an ABI 3500 genetic analyzer, and we used multiplexing in order to save on cost and allow various combinations of loci to be run together. The resulting electropherograms were manually scored by myself. Any failed PCRs were repeated at least once and then considered as missing data. All of the homozygotes and a subset of the heterozygotes were amplified at least 3 times to ensure accuracy of the scored genotype. Next slide. Diversity statistics for each locus and each population were calculated using microsatellite analyzers. Allelic diversity, observed heterozygosity, expected heterozygosity, and inbreeding coefficient were the indices that we looked at. We also tested for linkage to equilibrium between pairs of loci in each population, and we tested for Hardy-Weinberg equilibrium deviation at each locus per population combination using Fisher's exact test. We detected 3 pairs of linked loci, but that is likely an overestimate due to the presence of null alleles. 43 out of 60 population by locus comparisons were out of Hardy-Weinberg equilibrium following a Bonferroni correction, and this indicates, as expected due to the relatively recent founding of the southwestern U.S. quagga populations, that migration drift equilibrium is not yet established. In that same line of reasoning, no alleles were present in our data set, but they were also expected due to most loci being out of Hardy-Weinberg equilibrium. And thankfully, no genotyping errors were detected. Next slide. This table is summarizing the genetic diversity indices for our 6 quagga mussel populations. And we have them in order of priority here, not in alphabetical order. Lake Powell, Imperial Dam, Senator Wash, Lake Havasu, Lake Mojave, and Lake Mead. Visually, comparing the mean allelic diversity and allelic richness and the mean expected and observed heterozygosity and the mean inbreeding coefficient, you can see that there's very little variation among the populations. Next slide. This is the same table. The number of alleles per locus per population ranged from 7.0 to 8.9, with the exception of one locus, that's DBU92, and that locus exhibited only 2 alleles, which in looking back at the previous studies, they found the same thing with that locus. If we exclude DBU92 from further analyses, expected heterozygosity ranged from 0.73 to 0.75, and observed heterozygosity ranged from 0.48 to 0.54. All of the SIS values for all 6 populations were relatively high. Next slide. And that's the inbreeding coefficient, the SIS values. The microsatellite analyzer was also used to calculate both global and pairwise genetic differentiation among populations. Pairwise FST values ranged between 0 and 0.018. Our overall FST estimate was 0.0037, which indicates low overall genetic differentiation. Of the various measures of genetic differentiation, FST is the most conservative estimate. It's the most commonly used, and it's the best measure of genetic differentiation for microsatellite data. And AMOVA confirmed that there was no significant genetic differentiation among populations. We did, however, determine a relationship between genetic distance and geographic distance, indicating a pattern of isolation by distance, despite the low overall genetic differentiation. Next slide. This table summarizes genetic differentiation in the form of pairwise FST values among our 6 clogged muscle populations. Just a reminder to go across the top: Lake Powell, Imperial Dam, Senator's Wash, Lake Havasu, Lake Mojave, and Lake Mead. The pairwise FST values in the bold below the diagonal are the ones that are significantly differentiated. Next slide. The largest pairwise FST values were found between Lake Powell and Lake Mead, and between Lake Powell and Imperial Dam. The other genetically distinct population pairs include Lake Mead and Imperial Dam, and Lake Powell and Senator Wash. Next slide. Put these pairwise FST values into a graphical context, we use structure, which is a conservative estimate of genetic structure in the presence of null alleles. Structure harvester was then used to compare likelihood values across a variety of potential genetic cluster numbers. And an analysis of delta K was used to further assess the most likely number of genetic clusters in our data set. Next slide. We ran 10 independent structure runs for between 1 and 10 genetic clusters. The bar plot here shows the contribution of 2 putative genetic clusters across our 6 cloggle populations. While there was some indication that 2 genetic clusters exist, the representation of clusters does not appear to be geographically partitioned, and the best estimate for this data set is a single genetic cluster. And that's pretty obvious from looking at the bar graph there. Each little line is one individual, and you can see there's not a lot of mixing going on. Next slide. As we look back at the previous studies of cloggle population genetics, this table is comparing the changes in observed heterozygosity and inbreeding coefficients for 3 populations that have been analyzed multiple times over the past decade. While we're looking at this, I just want to note that different loci and different numbers of loci were used across these studies, which does make it a little difficult to compare. And some of the exact same samples were run 2 to 3 different times by different authors. In general, we see an increase in the inbreeding coefficient. You can see the SIS, our values, Lindsay et al. 2017, are a lot higher, actually, than the Marisco, and a lot higher than Brown and Stepien. And if we look just above that at the observed heterozygosity, it's just kind of a puzzling pattern there that changes over time. Higher, lower, higher, lower. Next slide, please. In this table, we're looking specifically at the change in allelic diversity by locus for the same 3 quagga populations across 8 loci that were analyzed by both ourselves and Janet. In the 4 to 5 years between these sampling events, we see a 2 times increase in allelic diversity across sites. And with the exception of 2 loci, we see a 1.2 to 3.5 increase across loci. Next slide, please. So to tie this all together, our observations of heterozygosity are similar to other Western populations, but lower than that has been reported for the Great Lakes populations. The main reason for this is that Marisco and Brown and Stepien used different loci in their studies. They used DBU 141 and DBU 110, which both exhibit high allelic diversity and high observed heterozygosity, whereas we used DBUG 2, and it exhibits high allelic diversity but low observed heterozygosity. Our measures of genetic diversity were higher than Genet's, but lower than Marisco's. The general increase in allelic diversity is most likely an artifact of our larger overall and per population sample size. A true increase in allelic diversity would occur through mutational processes, additional introductions from outside the region, or significant regional population mixing through gene flow. The time frame between the sampling events, though, is too short for mutation to be a significant contributor. And while we can't rule out regional dispersal, the differences are most likely due to sample size. It seems that the southwestern U.S. quagga populations are well isolated from the eastern populations by geographic distance, unconnected watersheds, and the watercraft inspection and decontamination regulations. Next slide. Overall genetic structure for southwestern U.S. quagga populations is on the low end, and only 1 genetic cluster was found. This lack of strong genetic differentiation is likely due to substantial initial effective population sizes or adequate levels of gene flow during the intervening years that have prevented rapid differentiation. Significant pairwise population differentiation was much less prevalent in our data set than it was for Janet, who reported high pairwise MST values with either 2 or 5 genetic clusters. However, her study was done prior to the infestation of Lake Powell. All reservoirs in her study made up 1 core population with mixed lineages, while the 2 pumping stations that she sampled were starkly different. With our study, we see that Lake Powell is the most significantly differentiated from Imperial Dam, Senator Wash, and Lake Mead, which has resulted in a pattern of isolation by distance. Next slide. For our final objective, to determine whether any genetic relationships exist with water quality measures, we note that Cucciarelli et al. Found relatively small quaggas in the southernmost reservoirs, that's Senator Wash and Imperial Dam. In these reservoirs, conductivity, temperature, and turbidity were all higher. While our results support 1 single genetic cluster in the region, one or the other of Senator Wash or Imperial Dam were included in 3 of those 4 significantly differentiated pairwise comparisons. Our combined observations of increased genetic diversity and increased inbreeding might support a scenario where currently undescribed environmental factors are influencing genetic mixing and individual reproductive fitness. But we'll have to keep looking because the population genetics of quaggas in the southwestern U.S. are still very dynamic. Next slide. Let me know if you have any questions.
Anybody have any questions for Denise in the room? No? Anybody on the phone have any questions for Denise? No? Denise, what do you think the future holds as far as genetic diversity of quagga in the whole basin? Is that one of those things that we would expect to have an increase, maybe from those water quality differences that you were talking about earlier, or do we think it's going to be a steady state type of thing, or? Hello, Denise? I'm sorry.
So we would think that if inbreeding was to continue to increase as it has,
Right.
we're going to see a reduction in genetic diversity over time.
Gotcha.
That may or may not be tied to morphological differences or water quality.
Interesting.
May or may not.
Well, I think that's one of the reasons why, I mean, my entire morning was filled pretty much with hearing these great people talk about containment and what kind of a monumental effort that is. And this is kind of one of those reasons why containment is so important, all right? We want to and need to maintain that relatively low heterozygosity, keep things fairly the same as we can, which will go into my next talk in a minute. But. All right.
Hey David, it also looks like you had a question on the messenger for the GoToMeeting as well.
Can you repeat that? I'm sorry.
It looks like on the messenger for the
I see.
GoToMeeting.
Thank you.
Heidi had a question on there.
All right. Let's see here. Heidi, you want me to answer? All right, Denise, can you hear this?
I can hear.
Heidi McMaster from Reclamation had a question. She said, or asked, based on this study, it appears that Lake Powell quagga mussel came from a different water source than the lower reservoirs. Am I correct in this observation? Sorry, I can't seem to get my mic to work. You want me to repeat it?
Can't hear.
I'm sorry. You want me to?
I would say no.
Okay.
But we don't have any evidence to support that. Right now, all we can say is that Lake Powell is more differentiated from those southernmost sites than it is from any of the other sites. So when it was infested later on, the infestation likely came from one of the closer reservoirs than it would have one of the southernmost. Or there could have been a genetic bottleneck. We didn't detect any, but they haven't been around that long. It's only been about a decade. And a lot of those things you just don't see in such a short amount of time.
Correct. All right. Did that answer your question, Heidi? You can type it in. I'll read it. I'm hoping. All right. Any other questions for Denise? All right. Thank you so much, Denise. I hope you feel better soon.
Thank you.
All right. Thank you. All right, let's go to.
So with this study that was done, will we be able to see if boaters come in from outside lakes or reservoirs and start to introduce new genetics?
Denise, did you hear that question?
No, I didn't hear. I'm sorry.
I asked if with this study, now that we have kind of an idea of the genetics here at Lake Powell, will we be able to see, I guess a little bit later on if, boaters or whatever start to bring in quagga mussel from other reservoirs as far as from a genetic standpoint, variation?
I think it's certainly possible if we did another study in another 5 or 10 years and saw some changes in the way that the populations are currently structured, then yeah, we could tie them. Populations are linked or not linked, and it would be interesting to see.
And I think increasing genetic diversity or increasing similarity in the populations, especially in the upstream, indicate.
Hey, this is Rick on Denise Amy. I think that's right. I think if you were to see increasing genetic diversity over time, or increasing similarity of the Lake Powell populations in downstream locations, that would be an indicator that there's probably movement upstream, which would a gene flow upstream, which would seem to only really occur if there were people moving, the veligers or the muscles around.
So would we, has there been enough studies done on genetic diversity of some of the reservoirs throughout the U.S. to where we might be able to pinpoint where it came from?
Yeah, we don't. And the populations are so similar. I mean, because the differentiation we saw between them is there was some significant differentiation, but it was still really, really low.
Gotcha.
So I don't think they're dissimilar enough to actually make that kind of fingerprint, make that kind of have a strong conclusion in that regard. So.
Okay.
All right, thank you both. So moving along, kind of in the same vein of what Denise just presented about. Last time I talked about the CRISPR-Cas9 system and gene drives and a gene drive specific for quagga, and I kind of wanted to pick up where we left off last time with that introduction. I had help with this. Melanie Culver's a conservation geneticist with USGS, has an appointment at the U of A. And Tom Deutschman works at the U of A. He's in charge of the genetics core. And they pretty much work on CRISPR-based technology 24/7. Not necessarily for invasive species, but for human health, especially things like autoimmune diseases. So they do CRISPR pretty much constantly. It's the basis of everything they do from now on. And I don't want to get too much into it because I did present this last time, which is online, but in case people haven't heard, CRISPR-Cas9 system, Cas9 is the enzyme. RNA are transcribed into short segments and guided to matching segments of DNA. So basically what you can use a guide RNA to pinpoint a specific segment of DNA, leave it out, put in whatever you want, and then have enzymes heal those ends. Right? So it's pretty new. Last, I forget the date, it was 2013 when it was first around, and it's a huge deal. Scientific breakthroughs come once every 20, 25 years or so. The last one we had before this was probably PCR. So this is pretty large. So when found that Cas9 binds to it and begins cleaving it, either turning a gene off or activating gene expression, depending on what you put into it. Right? It's like scissors. You go and cut it out, put what you want into it. What does the availability of that system mean for invasive species? Well, the nice thing is that you can create organisms in order to do what's called a gene drive. Okay? So you can use this super Mendelian inheritance of a transgene. Right? In normal offspring, you have a 50% chance of inheriting a given trait. Right? The offspring does, you have one of two subsequent generations. In a gene drive, virtually everything is forced to have that. They work, and they're very powerful, and they work relatively quickly depending on the organism. Okay? It's a very fecund organism that reproduces many times a year. Gene drives work relatively very quickly. If it's a longer-lived organism, not as quickly. Currently, I'll have to say that gene drives are probably our most powerful tool if we wanted to eradicate an invasive species. And even short of a full-scale eradication, we can make species or population far less fit. Okay? I mean, they're not magic bullets, and genetic diversity is one of those things that can essentially stop a gene drive. We're working with very small segments of DNA. So if there happens to be something in that wild population that doesn't pair with what we've inserted, then that gene drive's going to stop. So the more genetically diverse a population is, the harder it is for a gene drive to sustain itself. missing a targeted gene. That doesn't usually happen. It happened often with what we used to use, which were zinc finger nucleases. It doesn't happen that often with CRISPR. Of course, the big deal are social and ethical issues. Yeah, I mean, we're creating organisms. Okay? We're creating organisms genetically that we can put into the wild. We're hoping that they're going to die out, but there's always the degree of we're not quite sure. Okay? And one of the questions, of course, would be the potential eradication of entire species. And what's invasive here has a home range. It's like the drainage in Ukraine. So, is it possible for a genetically engineered organism to make it back someplace and affect the native population? Yes, it is. It's highly unlikely here in the Colorado River Basin. And I'll talk about why in just a minute. But, you know, a quagga or a gene drive for quagga probably wouldn't work in many other areas. Probably wouldn't work in the Great Lakes. I would have issues with it probably because a lot of those organisms that came here are capable of going back. Right? And we just talked a little bit about this geographic isolation. I mean, I can see getting from, you know, the Niagara River drainage, the Black Sea, into the St. Lawrence waterway and into the Great Lakes. We can see that. It's the Atlantic Ocean, ballast water, ships. It's right there. So large amounts of organisms are capable of actually going in there and being released all at the same time. What's more difficult, by a long shot, even all that journey across the Atlantic Ocean, was for them to get from there to the Colorado River. It happened, of course, so those things can happen. But compared to those two places, the Colorado River Basin is largely a landlocked area now. Largely. We have the occasional environmental flow. But are we always going to have those in the future? I don't know. Is water ever going to make it into the Gulf of California ever again? I don't know. So compared to the interconnectedness of the Great Lakes, drainage is everywhere. I mean, drainage is all the way in the Mississippi, Missouri. Colorado River is relatively isolated. And we also have what you all were talking about earlier today, and I was fairly impressed with, sophisticated containment efforts. Okay? So would it be possible for an engineered organism to escape the basin? Anything's possible. It would be unlikely. The highest incidence I think that you'd probably have would be in some of the California drainages because they're closer to the coast. But even there, gene drives often are targeted at things like fertility and fecundity. We'll talk about some organisms in a minute that have done that. The thing is, life wants to persist. I mean, it's, you know, genes targeting fertility and fecundity, those genes are very well conserved. I mean, things don't want to die. They want to persist. Okay? The CRISPR-based gene drive work by recognizing, like I said, those short genetic sequences. Individuals with differences in a particular segment would be immune to the gene drive. The lower the genetic diversity of a population, the greater the chance of success. That's why we talk about things like heterozygosity and homozygosity. Okay? Yet another reason why containment is important. We don't want more genetic material coming in from anywhere else. Just an example of a gene drive. Well, not a true gene drive, but the beginnings of one. 2015, researchers discovered a gene that causes an infertility mutation in female mosquitoes, Anopheles gambiae. Those are the ones that actually carry malaria in humans, to be passed on to all their offspring. Controlled experiments showed that the mutation increased in frequency over several generations. And it did. It's what you'd expect. Transmission rates are pretty high, 91.4 to 99.6%. Are those transfection rates high enough? Those would probably be high enough in something that didn't have the genetic diversity of mosquitoes. But even within that species, there's a large degree of genetic diversity, especially when we're talking about worldwide distribution just about. So resistance to that gene drive did emerge. These were in large mesocosms in a controlled environment. This was not out in the wild. So even in that small enclosure, it prevented some mosquitoes from inheriting that modified genome. And that's just in a relatively small area. Okay? And like I said, that resistance emerged due to the large amount of genetic diversity in that species. And I guess, Denise, are you still on the phone?
Yes, we're still here.
Well, as time goes by, I guess that was the base of my question. Will quagga in the Colorado River continue to exhibit relatively low genetic diversity? I guess I could say for the immediate future, right? Will that persist forever? I don't think anybody knows. It's already pretty low. I don't think it's going to go any lower than, well, I guess it could. But the more quagga expand their range within and adjacent to the basin, that's why some of those areas that are farther out have slightly different heterozygosity than the area, that kind of core population. The greater the chance for that heterozygosity to increase and bottlenecks to be alleviated. That's what we don't want. Right? That's why what you guys do is so vitally important. So instead of fertility and fecundity, we could potentially create a gene drive for more non-lethal attributes. Non-lethal doesn't mean it's something that's not going to affect or wipe out that organism over time. It just means that it's not fertility and fecundity. I guess my question is, can fish survive without gills? Probably not. Can birds survive without keratin formation from further? Can turtles lay eggs that can't biomineralize calcium? Probably not. That genetic variability, that problem with genetic variability is somewhat alleviated in these more non-lethal gene drives. So we're not looking at fertility and fecundity. The thing about CRISPR is that we don't have to necessarily, we can have multiple targets on any given strand of DNA or any protein or any gene. Okay? We don't have to cleave out and insert just one. We can cleave out and insert 1, 2, or 3 simultaneously. Some potential non-lethal genes to target for quagga. Obviously, the quagga have an absolute requirement for dissolved calcium, and a lot of the risk assessments for quagga and zebra mussels in certain areas are performed based on calcium concentrations in water. Right? That calcium requirement increases post-trochophore stage. In other words, when they start to form veligers, they start to form shells. Shells are made of calcium. Okay? Trochophore is something that we largely don't even think of. I mean, we're looking for veligers, right? Whatever size veliger happens to be out there. The trochophore is, it's a fertilized egg. I mean, it's right after that. They're not huge, 80 to 100 microns. You could still likely maybe see them with the naked eye. Not my eyes, but maybe somebody's. But the trochophore is an important stage in its life. Basically right after the fertilized eggs. And we sometimes see trochophores in phytoplankton samples, plankton samples. So what if we create quagga that had an inability to physiologically incorporate or biomineralize calcium? So we don't ever get to a veliger. They're just trochophores, and then they die. Okay? Veligers, 5 days. But for the life of me, I can't find out how long trochophores survive. Likely not nearly as long as veligers, because veligers at least have a shell of sorts to encapsulate themselves to prevent desiccation. Trochophores don't. They probably won't survive nearly as long as veligers will. And veligers are what we commonly associate with incidental transport. Not early trochophores. There is this protein called calmodulin. So in other words, we're not starting or trying to reinvent the wheel, or we're certainly not trying to figure out the entire genome of quagga. Previous work has shown us that calmodulin, it's important for basically every living creature for biomineralization of calcium. And we all have it, we all use it. Every carbon-based life form, pretty much. It's especially important in bivalves and mollusks for shell development. It's also dependent on other things, calP, calmodulin-like proteins. Both of those together are vital to shell regeneration and formation in all mollusks. Calcium-bound calmodulin forms a critical subunit for the regulatory enzyme phosphorylase kinase, which in turn is a regulator for glycogen breakdown, also binds and activates other kinases and phosphatases that play significant roles in cell signaling, ion transport, and cell death. So even if we didn't get to the point where they didn't have the capability to form shells, they probably wouldn't survive from those other things anyway. And like I said, we're not trying to reinvent the wheel. Calmodulin has been cloned from the Pacific oyster, other mollusks, bivalves, and the pearl oyster, and its function studied very recently. Okay? Also has been characterized to play an important role in calcium regulation in the freshwater mussel pearls. So we know, or at least we have an idea of its location and what it actually does. These calcium binding sites. It's also possible to create, along with that, a second tier of a quagga gene drive, development of byssal threads. Okay? They're completely dependent upon those adhesive plaque protein genes, or muscle foot proteins, which is what they form. MGFP2 is a major component of the terminal adhesive plaque of byssal threads. Could we actually simultaneously do something with that? Yeah, I mean, we could create an organism that never formed out of the trochophore stage, and by happenstance, if there happens to be something in the wild that had the genes that didn't pair up with that, they wouldn't be able to form byssal threads after that. So it's just kind of an added safety measure. Just some of the nuts and bolts. We'll go through this pretty quickly, but selection of those guide sequences at each gene and DNA replacement at those sequences, that's kind of the first step. Like I said, we're not trying to reinvent the wheel, so we'd use guidance from the previously published work. It's that CRISPR-Cas9 construct has been completed, transfection of that construct into the three different life stages, the trochophore, the veliger, and the petaveliger could occur. And we'd have to do that to determine the transfection efficiency of each post-fertilization phase. We don't have to do this in every phase. We want to figure out which one has the highest transfection efficiency and probably stick with that one, right? To determine which is the best phase to utilize it. So it's right there. And then we assess that resulting effect on those two systems, the calcium uptake and byssal thread function. Then we perform PCR to examine those indels, insertion and deletions, confirm they're where we want them to be, no missing parts. They're of the proper size for what we wanted to cut out and exactly what we wanted to replace, and to estimate genomic editing efficiency. Right? That's basically what we're trying to do. We can also use green fluorescent proteins that we can insert along at the same time. The nice thing about green fluorescent proteins is they fluoresce if the gene happens to be activated, right? So we can use fluorometry after that to actually look at how much that gene is activated or whether it's been activated or not. Final step is to add that gene driver to the CRISPR-Cas9 construct and then transfect again. That time we will look at the proportion of offspring inheriting the destructive traits over multiple generations. That would be our model that we could actually use to say, okay, here's how many organisms and the amount of time it would probably take for a gene drive to take effect. It's not that pie in the sky. It's really pretty, it's not easy, but it's not that difficult using everything that we can use today. And like I said, we don't need to know the entire quagga genome. It's not our goal. Just trying to create transfected organisms. For various reasons, and that's kind of a bolder statement than I wanted to make, but the Colorado River Basin, I think it is a viable candidate for a gene drive. I don't think other places are. Like I said, I don't think the Columbia River Basin is. I don't think the Great Lakes are. I don't think any of the Mississippi drainages are. But because of our geographic isolation, we're somewhat unique. So I think that's not a bad, it's a good thing for us in several ways. We still have relatively low genetic diversity, and we have those sophisticated containment efforts. We don't want things transported anywhere else. We want to, if possible, something after a lot of discussion, to implement a gene drive and then not have that material leave. We want it to stay here and hopefully just die out. The effects on other organisms, well, there'll be some. I mean, we'll go back to things as they were post-quagga, pre-quagga conditions. Okay? But because quagga mussel doesn't crossbreed with anything else, I mean, to a degree it does with zebra mussel, but those never form adults. And that is the closest thing that exists to quagga. So if even those can't provide survivable adults, then chances are it's not going to crossbreed with anything that's already out there, a native organism. And yeah, as far as I know, there's none known in the basin or even the world. And as that gene drive progressed, those concerns of containment would be lessened because we're not having to contain as many things. Right? Just to summarize, I do think the Colorado River Basin has unique aspects making it amenable to a gene drive that don't exist probably other places. It would be the most powerful tool that we know to control or largely eradicate, I don't know, quagga mussel in the basin. It's relatively inexpensive. It would be and that's just to create the organism. It's relatively inexpensive. Shouldn't directly affect any other organism or native mollusk. And should recontamination occur, we can do it again. That's it. Any questions?
So if you're introducing something that doesn't allow them to grow and mature, how do you perpetuate it so that it spreads throughout the ones that are already in the lake?
The F1 generation and F2, subsequent generations. Don't forget, initially they would be placed in there, adults, that carry that gene.
Yes.
So it's their offspring that would not be able to do that. And those offspring could potentially, well, they wouldn't mate with other things, but the offspring that those produced in the F2 subsequent generations would have that. They would not be able to produce calcium, they would not be able to form buccal threads, and over time, once we've introduced that genetic material into it would take over relatively quickly. I mean, quagga are 3 years till they're sexually mature.
Much quicker than that.
And so
how does that gene drive work? How do you get it to 100% of the descendants carry that gene?
because you've inserted something in there. I mean, you basically actually said, and I'm not a geneticist, that's why I'm working with no one in Culver. Denise or Rick, are you still on the phone?
Yeah, we're on the phone.
Can you, Mark here had a question, can you help him with it?
Curious about the gene drive and how it is that we managed to get all of the offspring having the gene we've inserted.
Yes, that's a good question. So, you know, from the mosquitoes, they typically are creating like a motherless gene drive, I mean a dogless population where all the offspring are male. And so that is able to perpetuate across the generations until you run out of females. If you're creating something that prevents all of the offspring, offspring from developing into mature adults, then you basically have to do like they do with the mosquitoes. That it's not necessarily a self-perpetuating gene drive; it's more of a CRISPR-driven genetic modification. That basically you rear up lots and lots and lots and lots of these muscles that have this gene drive on both chromosomes, this CRISPR-Cas9 construct on both chromosomes, and then you basically flood areas with these guys so that they soak up a lot of the mate, a lot of reproductive activity, and then don't have viable offspring, which radically reduces your population sizes. And you just have to keep introducing them until the populations go extinct, or you know, something, which is what they kind of want to do with what they've done with mosquitoes that have non-viable offspring through various mechanisms in different places here in the last couple of years.
So that F1 population, if it's reproducing with the muscles or whatever that's already out there, its offspring would be half and half having the new gene.
Right, except with the CRISPR-Cas9 gene drive, it's a selfish gene, and it copies itself onto the other chromosome at the same locus, at the same site where it occurs on the one that's been modified. So all of their offspring will have that gene because of that selfish gene behavior of the construct you put into the DNA.
Yeah, that's the bias in here in this aspect.
Fantastic! Wow, how powerful!
It's very powerful. That's what I'm saying. I mean,
yeah,
it can be a very powerful tool. Any other, yes, sir?
What does it take to get the ball rolling? Does it like take 20 years of input from whoever and all that stuff and signatures?
Well, I'm not sure what you're talking about. Let's just say, if somebody were to submit a proposal to the latest round of funding, in that proposal it would take about 2 years and about $300,000. Theoretically. Believe it or not, quagga, as invasive as they are not that easy to breed and to grow in aquaculture and keep alive in aquaculture. But we know how to do that. And that's one of the things that we do. We start with that population. And then working with Melanie Culver and the genetics core at the U of A, about 2 years to grow organisms, to actually transfect organisms, make sure that those indels are right where we wanted them to be, make sure that their the green fluorescent protein is doing what it's supposed to do, and then be able to come up to some determinations of possibly the number needed to put into the wild. So in a couple of years, and about that.
What are the negatives?
We got to make sure that they don't escape here. Here meaning here, the Colorado River Basin. Because we don't want to cause another species to go extinct.
That sounds like Jurassic Park.
Right. But we don't want to cause that. I mean, and like I said, if we go back to that map of California, the most likely places to get out to the open ocean would be in California. But even if they do, outside of being ballast water someplace else, could they survive an oceanic trip someplace? Probably not. Public.
Yeah, hey, this is Rick again. So the National Academy of Sciences recently produced a sort of an outlook on gene drives. One of the things that they recommend is that there are not just sort of physical barriers to them spreading to places where you don't want to do, like an ocean, right? But also intrinsic barriers, sort of the way that the gene drive is set up using like the CRISPR-Cas9 targeting technology. And there are in theoretical development now, things like that are called like daisy chain gene drives or daisy field or daisy form. And they are putting different components of the gene drive on different chromosomes and things like that. They're able to theoretically make it so that the drives are unlikely to spread beyond global area just based on genetic sorting and the potential for resistance to evolve. And so there's both intrinsic and extrinsic barriers to the thing spreading to places where you don't want it to be. And if there's something that causes all of the offspring to not be viable, then it really has a hard time spreading to someplace else because it's like you said, I mean, it's just all the offspring don't develop, so they really can't perpetuate that long in the wild.
Right. So we wouldn't have to, like I said, that's one of my slides going back, is that over time, as the gene drive progressed, we wouldn't have to worry about containment as much.
Having worked on them here and in the Great Lakes, I would think it would be hilarious if they all of a sudden ended up on the endangered species list for the Black Sea area.
Save the quagga.
Hopefully nobody's on the line from the Black Sea area.
Save the quagga. Yeah.
But we are very unique in that we don't have any native mollusks or that you have to do research to make sure whatever you're doing is going to impact them.
I mean, maybe something with a California floater, just to make sure, but as God is my witness, I have no, it shouldn't interfere with any other species.
Yeah.
It shouldn't.
So within the gene drive community, as you're giving a presentation like this, what kind of opposition are you seeing? And from who?
I've only given this once, and that was remotely. So this would be it.
So there's no friends of quagga?
Not that I know of. You guys would know that better than I would.
Yeah, they love the rats out on the Channel Islands, so we'd find some.
There will be, because it's, like you said, Jurassic Park-esque, it's going to be hard public sell. It would be. But at the same time, we create things all the time. I mean, in this particular case, in this particular basin, it's the safest area I can think of for a Dracaena gene drive.
So we've already done it with mosquitoes.
Well, they've done it with mosquitoes before. That was the fertility of the Kennedy aspect of it.
Was there any pushback on that?
They didn't do a gene drive with mosquitoes. They actually created the organisms and then kind of, am I right, Rick? Looked at them in mesocosms and determined that.
They created the mosquitoes where they have where all offspring were non-viable, so they had no offspring or at least close to no offspring, and they were able to do some deployments in South America and Central America and the Caribbean, and had really good success knocking down, at least annually knocking down the mosquito populations that spread malaria or whatever. But when they tried to do it in the States, they really hit a snag. People were just so afraid of genetically modified organisms that it's taken them a lot of effort to try and get buy-in down in the Florida Keys to do that. So yeah, so it's just that general distrust of sort of genetic modification is what you really kind of run into as your opposition.
I look at this from like a public involvement perspective when it comes to doing any sort of NEPA for a decision like this. And it seems like it could get kind of nasty.
Yes.
What if we could do it at a small 8-acre community fishing lake that doesn't go anywhere, that's an endpoint, and that water can be transported out to where there's no boating or kayak or any kind of access, that way it's only shoreline fishing.
Pilot scale?
In a city, it's actually a city resident too, but a pilot study to see if it would take us all the way. And then maybe find something for the apple snail. That would be it.
For the what? I'm sorry.
Apple snail, we gotta find something for it.
Sure. I mean, we could almost determine the same thing in the lab, though, that you're doing with the pilot study, but at least with the pilot study, you're kind of introducing it to the public, I guess you could say. I mean, that would not necessarily be so much from a data gathering standpoint, but somewhat. but from a public perception primer. Sure.
And maybe that would
And
help
Mark?
us go further.
Right.
From a public perspective or public perception perspective, a good maybe link to draw would be the screwworm flies work that was done in the 50s, because that was, they were essentially airdropping irradiated screwworm flies all across the U.S. That would seem like much more of a PR disaster than what you're describing here. So you may be able to try and link, this sort of work with something like that. And it's a very similar dispersal mechanism to what you're describing here for these for the GMO quag mussels.
Yes. Irradiated screwworms we don't want.
For anyone that isn't familiar, screwworm flies, they're sort of a parasitic fly. In the 50s, they airdropped a lot of them, sterile ones across the U.S. They only breed once when they reproduce, and then they die. So it was a pretty effective mechanism there. So if they couldn't reproduce in that first go, they died anyway. And then they were eventually driven back through Central America to Panama. So now we don't have problems with screwworm flies in North America. But this would be the dispersal system is pretty similar to what you're talking about here.
I agree. Yeah. Any other questions? All right, we're going to go ahead and wrap up. If anybody has any more questions for me, feel free to email me. I'm not sure when our next meeting's going to be. It may be a remote one, since it's supposed to be 187 degrees or something next week. So I may sit in air conditioning and do that one next. But certainly after that, I think we're probably going to go to Lake Havasu City, because we haven't been there for a while, and talk about issues in Havasu. But they're always going to be available online, as long as I have people that can help me with that. We'll always offer those go-to meetings. All right. Otherwise, I think we're good. Thank you, everyone online.
And thank you for doing this meeting here and sharing with us.
Thank you for hosting. This is a great venue. I like it. I will come up here anytime.
Okay. We are here.
Especially next week. If you could just have me up again next week, that would be great.